Hello, everyone. Thanks for coming today. It is my pleasure to introduce Dr. Michael Wendt from Purdue University. Dr. Wendt got his doctorate degree from the Medical College of Wisconsin in microbiology and molecular genetics. In 2007, he moved on to a postdoc here at Anschutz and worked under Dr. Bill Sheeman for 3 years and then continued to work under him for an additional 4 years at Case Western Reserve University. In 2014, he became an assistant professor at Purdue University, where I was fortunate enough to work for him for 3 years during my undergrad. And Dr. Wendt's lab focuses largely on the development and progression of breast cancer metastasis. And using a variety of different models, they investigate many processes such as EMT and the development of resistance to current therapeutics. And additionally, he works to identify novel therapeutic targets that could prevent or inhibit metastasis. So with that, I'll leave the floor to Dr. Wendt.
All right, thank you, Connor, for that introduction. It's really great to see a really productive student like Connor move into a group that I know personally is really well suited for him here at Colorado, and I hope he continues to be successful in his graduate career here. So also thanks to Tracy for the invitation. It's great to come back to Colorado. It's very surreal to see all the things that have changed. I took the train from the airport last night. That was very novel for me. It was always tough to get to the airport. But how many things have also stayed the same? And so it's great to see progress from a lot of PIs that were here when I was a postdoc, and to see people moving into new areas as well. So today I just want to take the next hour or so to talk about some of the recent updates from our lab. And so we titled the talk kind of broadly Pharmacological Targeting of Metastatic Breast Cancer. And so as Connor mentioned, we're very interested in the concepts around metastasis, how they feed into drug resistance, and why we can't seem to target or efficiently inhibit tumor growth once tumor cells have moved beyond the mammary gland into metastatic sites. And so just point out, it's the 150th anniversary of Purdue this year. We're just finishing up our 150th anniversary, and it's the 40th anniversary of our NCI designation as a cancer center at Purdue as well. And so we continue to look at breast cancer as a disease because it continues to be a highly prevalent, highly lethal disease. So the most diagnosed cancer in women, and the second most lethal cancer in women as well. And when we look a little bit closer at those statistics, we can really see that it is this process of metastasis that drives lethality in breast cancer, right? When women walk into the clinic and disease is confined to the mammary gland, survival rates are upwards of 100%. However, when those women walk into the clinic and are de novo diagnosed with de novo metastases, those survival rates drop to only 23%. And these are only 5-year survival statistics, right? And so a woman that has primary disease that's confined and local and metastatic disease is not detected has the opportunity or the possibility of walking back into the clinic years later beyond that 5-year window and now moving into that distant metastatic disease. So the concept of primary tumor diagnosis and these late or long-term relapses after a period of dormancy are something that we really don't understand in breast cancer. And we'll touch upon that a bit today. And so when we're thinking about metastasis, we really think about it as an entire process, right? It's a sequential cascade of several different events. And fortunately, that makes metastatic progression of disease very inefficient, if you will. And so that is good for the patients, but makes things more difficult to understand when we're talking about getting potentially genetic information or now diagnostic information from that primary tumor site. But really our pharmacological target, again, is organ systems away and time, again, years potentially away from those diagnostic materials. And so how do all of these processes, again, influence tumor growth, tumor characteristics in that metastatic microenvironment compared to the primary tumor microenvironment? And so there are obviously lots of things to focus on when you're talking about metastasis as a whole. If we have initiation of primary tumor growth as the first step, all the way from survival in the bloodstream, immune evasion, and then potentially the most rate-limiting step of metastasis is this concept of secondary tumor formation. If you think about a cell that's grown up essentially in a primary tumor that's very collagen-rich, very hypoxic, very acidic, now that cell is thrust into the bloodstream, circulates through the body and might find itself here in this situation, for instance, in the lungs, right? A completely different microenvironment that's oxygen-rich, very fibronectin-rich, very compliant. How does that same cell that survived all those stressful situations now reinitiate the growth program? So you can really, I hope, appreciate the difficulty that a tumor cell endures to complete the metastatic process. And so it's not surprising, again, that these secondary tumors are quite unique from the primary tumor. And when we think about metastasis, we think about the process of epithelial mesenchymal transition. And so this, largely at its core, suggests that you have one cell lineage, a differentiated epithelial cell that gives rise to a carcinoma, changing that cell lineage into something that's not epithelial, right? Something very nondescript as mesenchymal. And whether or not a change in cell lineage actually happens is a conversation that I love to have with people, whether or not an epithelial cell can actually change into a fibroblast. But for the purposes of today's talk, we're really more interested in these transient changes in phenotype, again, that allow a tumor cell to complete these steps of metastasis. How does a transient change into a mesenchymal phenotype allow a cell to survive in the bloodstream, allow a cell to evade the immune system, and then allow a cell to adapt to that secondary microenvironment only to change back to a proliferative phenotype? And so this reversion back to an epithelial phenotype known as mesenchymal epithelial transition is simply that. However, we like to think about it in not a absolutely conserved reversion process. So again, if you have an epithelial cell in this microenvironment, now it's moved to a different microenvironment. When that cell redifferentiates, if you will, back into an epithelial phenotype, how does it take in those microenvironmental stimuli to potentially change to something that's different? So very analogous to a developmental process. And finally, the processes of EMT and MET don't take place all at the same time, right? When people think about EMT and MET, a lot of times it's very easy to focus on one cell completing this entire process. But actually, you have tumor of multiple cells, right? And they're completing EMT and MET at different rates. And so this concept of plasticity naturally feeds into the concept of epithelial mesenchymal heterogeneity. And now how do these two cell types, one being more like a fibroblast, one being more like an epithelial cell, how do they talk to each other? And how does heterogeneity contribute to tumor progression, drug resistance, and metastasis? And so overall, we can term that epithelial mesenchymal heterogeneity. And so today, we'll keep coming back to this schematic in which we have an epithelial cell here characterized by cell-cell junctions, transitioning to a mesenchymal cell characterized by a more dynamic actin cytoskeleton, expression of more matrix proteins, expression of matrix degrading enzymes, and other factors that we'll talk to. And so the first question is, can we target mesenchymal cells? We don't spend too much time in the lab looking at inhibiting this EMT process because potentially that's taking place very early in tumor progression, right? Cells are known to disseminate very early, even before primary tumors are palpated or diagnosed. Secondly, if cells move beyond this mesenchymal state and are now adapted to that microenvironment, and they must re-engage an epithelial program to form a macroscopic metastases, can we inhibit this reverse process? And would that be capable of delaying macroscopic tumor growth, right? If a cell has disseminated, it's undergone EMT, and if it does have to revert, which lots of controversy around that topic, but if it does have to revert back to an epithelial phenotype to form a macroscopic metastases, can we delay that process to allow disseminated cells to maintain themselves, but maintain themselves in an asymptomatic state? And then the last topic we'll touch upon, if I don't talk for too long, will be this concept of what is different between these two epithelial states. So again, if you have an epithelial population in a primary tumor that has undergone EMT, has undergone MET, has changed its microenvironment, how do these two epithelial states differ from each other? And can we use that information, again, to derive pharmacologies that are specific to metastatic tumors? And so if we're going to focus on this concept of EMT and metastasis and MET, we really need to start with some type of material that's characteristic of this primary epithelial state, right? If you just take a cancer cell line off the shelf or from the ATCC, these are all derived from spontaneous mouse models or cultured from human tumors, and they've probably been through this process of EMT several times. Several cell lines are derived from metastases, right? So all of these processes has already happened, so you don't really have a control or starting type material to compare to. And so we spend a lot of time in the lab building transformation models. And so we're very interested in this ERB family of signaling molecules. We built a lot of transformation models using EGFR, but for the purposes of today's talk, we'll focus on HER2, right? And so just to remind us all of our molecular biology, EGFR and HER2 are part of this ERB family of signaling molecules, ERB1 through 4, and they can homo and heterodimerize to increase the diversity of ligands that those receptors bind. And HER2 in particular is overexpressed and genomically amplified in a large percentage of breast cancer, really defines this HER2 subtype of breast cancer. And because of that, several therapies targeting this growth factor receptor has been developed and are clinically utilized, such as antibodies, trastuzumab and pertuzumab. There's an antibody drug conjugate called trastuzumab and tanzine that we'll touch upon a bit. And also kinase inhibitors to inhibit the kinase of this receptor, kinase activity of this receptor. So here we show lapatinib, and we also have second generation molecules such as neratinib and afatinib. And so we were able to take this cell line, and so if you always look, we use a lot of different cell lines in the lab, and so we'll go back and forth between some of them. In today's talk, if you always just look here in the upper left-hand corner, I always try to denote what cell line is being used on that slide. And so here, we've taken this HMLE cell line. It's a pretty standard cell line, human mammary epithelial cells. And we can overexpress wild-type HER2 in these cells, and that's sufficient to get tumor growth. So here we just show a HER2 overexpression, and when you engraft the control HMLE cells onto the fat pad, these mice don't form tumors. So a prototypical immortalized, non-transformed cell line. But again, just overexpression of the wild-type receptor gets us tumor formation. If we look at those histologically, it gives us a very nice representation of what we see in patients, right? So this is a histological section of a HER2 patient stained for the receptor, and we see nice stromal involvement, and we can recapitulate that again in this mouse model. In these cells, as we want, or these tumors, are not metastatic, right? We get nice tumor growth and formation, but we have never seen metastasis in a mouse bearing these tumors. Moreover, we can take these clinically used drugs, lapatinib or the antibody drug conjugate, TDM1, and treat these tumors and very efficiently inhibit tumor growth, right? So here's our normal tumor growth rate. We can inhibit that with a kinase inhibitor, and we can even regress tumors, again, using this powerful antibody drug conjugate. So definitely a model of HER2 transformation, but getting back to this concept of EMT and epithelial mesenchymal plasticity, one of the first members of my lab, a postdoc, Wells Brown, was very interested in looking at differences in EMT stimuli and how that allowed those cells then to metastasize and how those EMTs were regulated in those cells. And so he began stimulating these, what we call HME2 cells now. So these are the HMLE cells that are transformed by HER2 overexpression, stimulating them with different things to drive EMT. And so here we show TGF-beta is a cytokine that I've been involved with for several years now, is one of these prototypical inducers of EMT. And so if you take the HME2 cells and treat them with TGF-beta, it takes a long time. So this is kind of an interesting model in which TGF-beta can induce EMT in some cell models in a matter of days. In these HER2 overexpressing cell lines, it takes nearly 4 weeks before we really get a robust EMT. And we can kind of see that here if the resolution is good. And we can also get an EMT by treating these cells with lapatinib, that HER2 kinase inhibitor, right? So the HMLEs grow in a defined media. When you overexpress HER2, you can take them out of that and grow them in normal FBS. But because of that, they're very dependent on HER2 for pro-growth signaling. And so if you treat with lapatinib, you see potent cell growth inhibition for several weeks, but eventually you get a mesenchymal population to emerge. And so 2 different stimuli, 2 similar morphological changes. The trained eye can tell them apart under the scope. So differences right away. But what was more important is when we take that stimuli away, in terms of the TGF-beta, over a 14-day recovery period, we can see these epithelial pockets of cells start to reemerge. And so if we just stain those cells using CD44 and CD24, 2 kind of prototypical markers of epithelial and mesenchymal cells, we can see this heterogeneous culture that forms after treatment and recovery from TGF-beta, right? So here are the parental cells. They're 44 low, 24 high. If we treat with TGF-beta, all of these cells move into this 44 high, 24 low population, but then upon recovery, now we see this heterogeneous culture. And so this is what I was referring to about how epithelial mesenchymal plasticity and this concept of EMT and MET taking place at different rates naturally feeds into driving heterogeneity. But in contrast to TGF-beta, the EMT we get with lapatinib resistance, what we call these LAP-R cells, is very stable, right? So here we take the lapatinib away, and this is at 2 weeks. We can culture these cells for several passages, and they really maintain this mesenchymal phenotype. So a little bit different than the TGF-beta if you look at the 44-24 profile, but again, the importance being that it's very stable. And we can culture them, and they maintain the resistance to lapatinib. And so we can just see that here. If we take the parental population, we see a nice dose response to increasing concentration of lapatinib. If we take those lapatinib-resistant cells, they're very resistant. And the first question we wanted to ask around these kind of two different models of EMT is a cytokine-induced EMT sufficient to drive drug resistance, right? So we have selection in a drug driving EMT, but is a more physiologic induction of EMT using a cytokine, TGF-beta, is that going to be sufficient to drive drug resistance? And at least in this model it is. We can see that we have a highly significant difference in this dose response to lapatinib. And if we take a little bit closer look at those cells, again using a 44-24 flow, we have this mixture of mesenchymal and epithelial cells here following TGF-beta treatment and recovery, what we call post-TGF-beta. And if we treat with lapatinib, we're reducing that epithelial population and enriching for that mesenchymal population. Whereas here, with lapatinib treatment here over 96 hours, we really don't affect the mesenchymal status of those LAP-R cells. And so Wells was very interested in these phenotypes and went ahead and flow sorted these populations here, highlighted by the red circles, where we have the parental cells, the mesenchymal cells induced by TGF-beta, and the mesenchymal cells induced by lapatinib treatment, right? And so now we wanted to conduct gene expression analysis to understand the differences and similarities between these cells. And so both cells were resistant to lapatinib, and so again, the first question we were interested in is this quadrant here. What's similar about these cells? So here we have gene expression for the parental cells on the ones, if you will, on normalized to 1 on these axes. And then we have gene expression changes in the lapatinib-resistant cells here, and gene expression changes with TGF-beta here in these sorted populations, right? So we're just purely looking at the mesenchymal populations here. And overall, you can see that this correlation is quite high, right? These two EMTs very similar, right? Morphological change is similar. Lots of gene expression changes are similar. And so we focused really on these most upregulated genes that were similarly regulated in these two models. And so a lot of the EMT players that you would expect are there. Twists are up, snail is up, zeb is up, all of these prototypical transcription factors that feed into this mesenchymal phenotype. As novel drug targets, we began to focus on FGFRs. And so here we see a potent upregulation of FGFR1 and FGFR4, and also upregulation of their ligand FGF2. Right? And here we're just recapitulating those findings using RT-PCR. So we have a HER2-driven model of cancer. When it acquires resistance to molecules that target the activity of that receptor, we have upregulation of this alternate RTK, or this bypass signaling pathway in FGFR. And so because of this and a lot of other research going on in the lab, we began to collaborate with our medicinal chemistry colleagues, and here this is in collaboration with Nathaniel Gray and his postdoc Lee Tan, to make these covalent kinase inhibitors of FGFR. So this is a molecule from Novartis, BGJ398, which was really one of the first FGFR-specific targeting molecules. The medicinal chemistry community struggled for a long time making a receptor tyrosine kinase inhibitor that was specific to FGFR, but BGJ was one of the first, and so Lee was able to put these adducts on that scaffold, allowing this acrylamide group here to form a covalent bond with a cysteine residue that lies just outside the ATP binding pocket in FGFR. And so just using a simple phospho-ERK signaling assay here, we can take cells and treat them with FGF2, the ligand, and see a nice induction of ERK signaling, and we can see that that's potently blocked by BGJ as well as these covalent kinase inhibitors. And so now if we go back to these flow cytometry assays and treat cells now either with lapatinib or our FGFR inhibitors, our covalent inhibitors, we can see we're targeting two different cell populations. And so this was an exciting day in the lab for Wells. He came back with this flow plot showing that lapatinib is efficiently reducing the epithelial cells, whereas the FGFR inhibitors are efficiently reducing the mesenchymal cells. And so obviously if you take these two molecules and you combine them together and do a cell viability assay, we can see again the post-TGF-beta cells resistant to lapatinib, whereas the parental cells are more resistant to the FGFR inhibitors, and we combine them together, we see this additive effect. And so there's an important point. I wanted to show these data for students because a lot of times students, when you start thinking about pharmacology and combining two drugs, right, you're focused on two different signaling pathways and a bypass pathway possibly driving resistance. And a lot of times people focus on things happening in the same cell, right? But it's important to understand your culture and what you're working on to really get a concept around when you put two drugs together, you might actually be targeting two different populations of cells. And so around this time, Saeed Akhan joined the lab and liked these data, but thought they were pretty academic in that he came to me and, you know, breast cancers are treated not necessarily with lapatinib right away, but they're treated with trastuzumab, this antibody, and this antibody drug conjugate, trastuzumab and tansine. And so, you know, we might have cells acquiring resistance to lapatinib and sensitivity to FGFR inhibitors, but they all still overexpress HER2, so they're all still going to be sensitive to trastuzumab and tansine, because as long as that antibody can bind to cells and take up that microtubule inhibitor, they're going to be targeted. And so Saeed was able to take either the post-TGF-beta cells or the parental cells, treat them with TDM1, and over 2 weeks, we see a very potent response, right? This is a very potent, very cytotoxic drug. And so we nearly clear the plate after 2 weeks. And so this is different than what we saw with kinase inhibitors, right, where the post-TGF-beta population was immediately resistant. So now Saeed sees that it takes 5 weeks for resistant colonies to emerge. And that's the difference between TGF-beta and the parental cells, is that in the parental cells, we don't see acquired resistance to TDM1. But with a post-TGF-beta population, we don't see immediate resistance. But now we have EMT facilitating acquisition of resistance to TDM1. So something a little bit different than what we saw with kinase inhibitors, but still therapeutically relevant. And in fact, when we take these cells and grow them up, we call them the TDM1 resistant cells, or TDM1R, we can see that in fact they have lost HER2, which is quite surprising, right? Because this is a cell model in which we artificially overexpressed HER2 using a retroviral approach. And so the ability of these cells to downregulate this molecule in response to that therapeutic pressure of TDM1 is really quite remarkable, and we can see here by prototypical markers of EMT that we're really selecting for a highly mesenchymal population that's ECadherin negative, NCadherin positive, and highly positive for vimentin, and our FGFR expression is further enriched, and if we treat those cells with our FGFR inhibitors, you know, not a great sensitivity, not like we saw with the lapatinib-resistant cells, so something else going on in these cells, but we are able to flip the sensitivity, in which if we look here, the parental cells, they're not sensitive to an FGFR inhibitor because they don't really express the receptors. But now we have a TDM1 resistant cell. These cells are highly resistant to lapatinib, don't express HER2 anymore, and so now they actually become more sensitive to an FGFR inhibitor. So that's exciting for us. And so, you know, Saeed's paper, we just put it on bioarchives. Hopefully it'll be out soon. Went on to look at some more mechanistic studies around this. He's able to push this forward using some more preclinical models. Here's a PDX model from Elena Whelm's consortium in which this was a HER2-positive patient that failed on trastuzumab. So we can take these samples, grow them up with direct transfers in mice. And so these are studies that Connor was highly involved in. And if we treat those cells with TDM1 or those tumors with TDM1, we really don't see a response. But if we then switch those cohorts of mice to an FGFR inhibitor, now we see this nice inhibition of tumor growth. And so when cells lose HER2 expression, they really tend to switch subtypes. And so I'm not a huge fan of breast cancer subtyping. A lot of people are, and it's very important and obviously drives therapeutic indications for the HER2-positive subgroup. But when you start thinking about metastasis, cells and tumors definitely have the ability to shed these kinds of drivers. And so there's well-documented clinical evidence of HER2-positive tumors becoming discordant, where they no longer express HER2 in the metastatic setting. And so essentially they're able to switch subtypes. And so if we lose HER2 expression, that's essentially recapitulating a triple-negative phenotype that's not expressing ER-PR and not amplified for HER2. And so we've been looking at FGFR inhibitors in the triple-negative setting as well. And so here again is a PDX model in which we expand these tumors onto several mice, and here now directly treat with our covalent compound and see a very nice inhibition of tumor growth. And if you're going to look at targeting metastasis, probably the best model to look at, in my opinion, is the 14-1 model, right? If you're really going to look at metastasis, and this is something that you want to pharmacologically inhibit, you need to get cells into a metastatic setting. And the 14-1s are really a robust model because you can put a primary tumor on mice, allow that to grow up for a couple weeks, and then you can do a surgery and remove that and allow those mice to recover for whatever time period you want to. And pulmonary metastasis will take place in 100% of those mice. And that's really the power of the 14-1 model, right? If you want to look at targeting metastasis, you need a model that metastasizes 100% of the time. A lot of models claim to be metastatic. A lot of models are metastatic, but not in 100% of animals. And so if you're trying to inhibit that process, it can be very difficult if you don't have a robust model. And so here, if we do that with the 14-1 models and then do surgery and only treat after metastases are detected, right? So this is day 1 after we started treatment. We can see nice detection of metastasis by bioluminescence here. We can nicely prolong or delay pulmonary metastatic tumor growth and prolong survival in these animals. Another model we use a lot in the lab is the 4T07s. So the 4T07s are kind of a sister cell line to the 4T1s. They came out of the same tumor, developed by Fred Miller's lab. And the 4T07s are quite different from the 4T1s in that if you put a primary tumor on the fat pad, you can grow that up. If you do a surgery, those mice won't get metastases. Numerous labs have identified micrometastases throughout those mice, though, suggesting that those cells can disseminate, survive, but again, they fail at that last step of metastasis. They're not able to re-engage a proliferative program. And so we're really interested in the lab around these concepts of what holds cells in this kind of state of dormancy or latency, if you will, after they disseminate. And so the 4T07s are a great model for that. And so Saeed has done a lot of work with the 4T07s, and here is just kind of the cutthroat experiment, really showing that this dormancy phenotype of the 4T07s is indeed dependent on the immune system. Right? If you do the same experiment in a nude mouse, those mice will all go on to get metastases. So the dormancy phenotype of the 4T07s depends on immunity, has to be done in a syngeneic transfer model. And here kind of the ultimate experiment was to put a primary tumor on, allow them to grow up, do a surgery, and then only at that point, after those cells have disseminated and you remove the source, now say you deplete CD8 positive cells using an antibody approach, about 60-70% of those mice will recur with metastatic disease. So really suggesting that CD8 positive immunity is required to maintain the dormancy phenotype of the 4T07s. And so getting back to this concept of FGFR inhibition, if we take 4T07s, and unlike the phenotype you get when you put them on the primary tumor, if you directly load them through the tail vein, we see robust tumor growth in the lungs, right? And so here, if you load cells in the tail vein, you usually have to sack those mice around 16-20 days because of robust pulmonary tumor burden. And so here Saeed is loading cells in the lung, waiting 2 days, and then initiating FIN4 treatment or FGFR treatment every day, oral at these different concentrations, and sees very nice, potent inhibition of pulmonary tumor outgrowth. And so looking at these tumors a little bit closely, again, we knew that these phenotypes around the 4T07 are very dependent on immunity. We can do classic immunological experiments where you put primary tumors on and you cut them off and then re-challenge with cells, and cells acquire immunity. So a very immunogenic model. And so looking at these pulmonary tumors that form, we can see they're able to do that because they're able to exclude T cells. And so if you can kind of see here, this is a stromal tract with CD8 positive T cells here by 2 tumor nodules. And they're very efficient, again, when loaded into the tail vein at very high numbers, at excluding T cells. And potentially that's why they're able to grow in the pulmonary microenvironment via that delivery mechanism, as opposed to being able to spontaneously metastasize from the fat pad. But in any event, when Saeed looks at those tumors for CD8 positive staining, FIN4 treatment, or again, FGFR inhibition, very nicely increases CD8 positivity in those tumor cells. And so obviously, a lot of work these days centers around checkpoint blockade and immunotherapy. And so this concept seems quite obvious at this point, right? If we're drawing in more T cells, can we now add in a checkpoint blockade therapy here in terms of blocking this inhibitory interaction between PD-1 and PD-L1 to further enhance an anti-tumor response? And so this is a nice quote from a recent paper that said among 36 variables, estimated T cell positivity is the number 1 predictor of response to checkpoint blockade. And so through a lot of experiments, which I don't have time to talk about today, Saeed was able to optimize, Saeed and Connor were able to optimize a sequential dosing approach in which you pre-treat with an FGFR inhibitor to draw in those T cells, and you need to stop that FGFR inhibitor to prevent inhibitory activities on T cells. But if you do that and then treat with an anti-PD-L1 checkpoint blockade, we see a nice prolongation of survival with our FGFR inhibitor, as we've seen before. And now if we add in PD-L1 after that, we can very nicely further extend that survival of those mice. And again, this tumor model, PD-L1 alone, no effect because there's no T cells in those tumors. And so this concept, fortunately for the science, maybe unfortunately for Saeed's manuscript, is definitely moving forward clinically. A very nice paper from the Janssen group, or the Johnson & Johnson group, that developed this FGFR inhibitor, or ratifitinib, which was just FDA approved in the spring, very nicely supports these kind of concepts of combining an FGF inhibitor to draw in more T cells and then combine that with checkpoint blockade. And indeed, there are clinical trials in hepatocellular carcinoma, in urothelial carcinoma that has FGFR3 activating mutations, combining, again, other FGFR inhibitors with checkpoint blockade. And so overall, the lab is very interested in FGFR. I could talk for all afternoon about FGFR. Where, in breast cancer, I think things are a little more complicated. We have FGFR1 being amplified in breast cancer, and about 13% of breast cancer patients will be FGFR1 amplified. Obviously, we have this upregulation of FGFR through the EMT process, but really we don't have a good grasp on how to apply FGFR inhibitors in cancers. And these other examples, right, we have activating mutations that take place in FGFR that drives, those diagnostics drive indications for FGFR inhibitors. So we're very interested in developing biomarkers around differential splice variants, co-receptors, changes in subcellular localization that might predict for patients that would respond particularly to FGFR inhibitors. I would say at this point there is a plethora of kinase inhibitors around FGFR. We're definitely probably out of the FGFR inhibitor game at this point, but still very interested, obviously, in combination therapies and other approaches to target the receptor. So, getting back to this concept of targeting epithelial mesenchymal plasticity, if we have cells that have moved into a mesenchymal state, we really think that FGFR inhibitors are a good candidate. But if cells have progressed beyond that, right, and are established at disseminated sites, can we prevent their transition, again, back to an epithelial state in the hopes of delaying or preventing metastatic tumor recurrence? And so here, if we go back to this gene expression analysis, right, before we were interested in what was the same between these two models of EMT, either TGF-beta or lapatinib resistance, now we're really interested in what's different, these two quadrants here, right? And so again, we had these RNAs isolated from these either parental epithelial population or these two mesenchymal populations. So a new student in the lab, Sebastian, is doing single-cell RNA-seq on some of these populations, and we'll see what comes out of that. But we did bulk RNA sequencing analysis, and nothing really jumped out at us about why a lapatinib-induced EMT was not reversible, where a TGF-beta-induced EMT was reversible. And so another assay that we did to characterize these cells is this substrate-based kinase activity assay. And so this was in collaboration with Chris Wiley at the University of Alabama. And so here you take lysates from those cells, and they spot them out over these arrays. It rinses them up and down over these defined peptides that are known substrates for particular kinases. And you can use a phosphospecific antibody to detect how much those peptides get phosphorylated and extrapolate that out to differential kinase activity in your sample. And so one thing that came out of that was a slight increase in this molecule spleen tyrosine kinase, or SYK. And so SYK, that data really jumped out at me because SYK has been known for a long time to be a very nice marker of EMT, but its expression doesn't go up. its expression potently goes down. And so we can see that here by RT-PCR, or this is actually the RNA sequencing analysis, that Syk expression is down in both models of EMT. So here we call the 44 highs, or those post-TGF beta cells. So to have a slight increase in kinase activity of Syk in these samples was really quite surprising, because expression of the kinase is going down so potently, right? And so here we can see the protein's going away, and we can't really detect phosphorylation of the protein, potentially because these levels are so low. And so if we take the kinase activity data and normalize that to the mRNA expression values, we can see that the post-TGF beta cells have this potent upregulation of kinase activity on a more per molecule basis, right? And so that's really the drawback of these kinome kind of assays, is they're really not normalized to anything, right? You're just looking at raw kinase activity. And so here now, when we kind of take this crude approach of using mRNA expression as the denominator and kinase activity as the numerator, we can start to appreciate this increase in kinase activity per molecule. And so to get a better grasp on this, we collaborated with Casey Kruzmark at Purdue. And so Casey is a by training a synthetic organic chemist, but really is interested in conjugating molecules to oligos, right? And so he does this a lot with small molecules. For instance, you can do complete small molecule screens in one tube because you use your DNA barcode as your readout. And so in this assay, he's conjugating what he calls Syktide, is an optimized peptide substrate for spleen tyrosine kinase, and he conjugates that to this oligo. And so you take that and you mix it with lysates from your different samples. So again, here we have the parental cells, the TGF-beta-induced mesenchymal cells, the lapatinib-resistant cells. And you pull out, you mix the peptide oligo conjugate in there, and you pull that down with a phosphotyrosine antibody. And now here your readout is done by PCR and sequencing, looking for, this barcode. And by doing this, we can really see this increase in Syk activity in TGF-beta-induced cells. And so, again, I think for the students, this is a great example in which the cancer biology community is completely infatuated with changes in gene expression, right? RNA sequencing analysis, changes in mRNA, all of these databases now are largely mRNA-based. And so it's something to think about when you're thinking about novel targets. If they're going to be enzymes, it's not really the expression that drives importance, right? We're interested in enzymatic activity. So things like HER2 are quite unique. And when HER2 expression increases, it forms these oligomers that auto-signal, right? So a lot of kinases, their expression and their activity are not necessarily correlated. In any event, Syk was very exciting because it also is a validated pharmacological target. And so there's this molecule fostamatinib that's a prodrug that gets dephosphorylated here. And so when we use it in vitro, it's R406. But the drug that was recently FDA approved, again, is fostamatinib, and it's approved for an autoimmune disease. As the name suggests, spleen tyrosine kinase is highly expressed in immune cells, such as B cells and macrophages. And so this inhibitor is used to prevent some autoimmune effects that take place on thrombocytes. And so now this is kind of, again, I won't go too deeply in this. It was recently published in the spring, but this was kind of the take-home experiment for me, in which, again, we take those HME2 cells. If we treat them with TGF-beta for 4 weeks and allow them to recover, we see these regain of this epithelial population, right? And we can, again, characterize that by a simple 44-24 flow. We're here immediately 4 weeks after treatment. All of those cells are mesenchymal. But after a 2-week recovery period, we start to see the epithelial population. And if we add the Syk inhibitor during that recovery period, we don't see the return to that epithelial phenotype. So just as we're hoping to do, we're stabilizing that mesenchymal phenotype. And so again, we go to the 14-1 model, do that experiment where we put tumors on, do a surgery, and only after that surgical intervention do we treat those mice. And in doing that, we can nicely delay the metastatic outgrowth of 14-1 cells. And here we just look at them histologically. 14-1s form these big nodules at this time point, and those mice treated with fostamatinib. Again, we're not changing dissemination, right? We haven't even applied the drug until after the cells have already disseminated. They're in the lungs. Here we're holding them in this mesenchymal state and delaying macroscopic tumor formation. So FGFR inhibitors targeting a mesenchymal population, fostamatinib, delaying MET. So our last topic will be, what is unique about this secondary epithelial phenotype? So again, cells are in one microenvironment of a mammary gland tumor. They're disseminating and growing up in a very different microenvironment, whatever that metastatic destination is. And so if we go to this kind of our reservoir of hypotheses, if you will, this HME2 model, if we look at these cells, we're able to get material derived from the parental cells versus this metastatic population. And so again, if we take the HME2 parental cells, we put them on the fat pad, we get tumor growth, we have never seen metastases. If we artificially create heterogeneity, at least in this model, by mixing the parental cells with the lapatinib-resistant cells, right? So now we have two, kind of a constitutive epithelial model that we know doesn't undergo EMT in this context, and we mix that with a mesenchymal lapatinib-induced model. That doesn't get us metastasis in this model. But if we take these cells, again, that have been treated and recovered from TGF-beta, that are now plastic and heterogeneous, we do see metastases. And so these metastases we're able to get out of the long bones of these mice. We call them the HME2 bone metastatic cells, or these BM cells. And if you culture them, to look at them under the scope, they look exactly the same as the parental cells. By 44-24, we can see they're slightly different, 24 low, but not 44 high. So definitely, again, if these cells either have metastasized or have undergone EMT, they've reverted back to an epithelial phenotype, or potentially this could be metastasis from the epithelial population. And so Sebastian in the lab, again, is our bioinformatics expert, took the RNA expression values from these cells as well as the TGF-beta-induced mesenchymal population, right? So this is a lot of data to look at here, but if we can see by this dendrogram, we can see that the parental cells, these two middle columns, are very much related to the bone metastatic cells. And several clusters, such as cluster 4 here that he identified, are your classic EMT genes, right? So these are genes that get upregulated with TGF-beta, and then when cells return to an epithelial phenotype, they go back down and return to baseline, if you will. And so Sebastian of late has been very interested in this cluster 1. These are genes that are not regulated with EMT. We can see they stay at the same level in the mesenchymal population compared to the parental population, but they only become upregulated in this secondary epithelial state, in these BM cells, right? And so there's several genes listed here. We've been very interested in the function of this molecule transglutaminase 2. And so what is transglutaminase? It is otherwise known as meat glue, is used commercially because it's a cross-linking enzyme. So if you like imitation crab meat or any kind of loafed meat, they take those meat proteins and mix them with transglutaminases, and that cross-links the proteins together to get you this loafed effect. But transglutaminase 2, very important molecule in cancer biology as well, as it acts in this cross-linking mechanism, linking amino acids together, particularly in the extracellular matrix. And so if we just use RT-PCR, we can confirm those RNA sequence analyses, again, where we don't have regulation of TGM2 during TGF-beta-induced EMT, but it only becomes upregulated in the secondary epithelial state, or these bone metastatic cells. And so again, here, if we look at fibronectin going up as a classic EMT marker gene, ECAD going down, TGM2 not changed until cells revert back to an epithelial state. And so here, ECAD goes back to pretty much the same level, whereas fibronectin does remain slightly elevated in this model. And so that really paints the picture in which now you have a cross-linking enzyme and potentially more production of matrix. If we take those two proteins, we can very nicely predict for changes in patient survival based on their expression levels. And so if we just knock it out in the 4T1 model, using a CRISPR approach, we can very nicely delay primary tumor growth, but also delay metastatic progression. And so, data like these are nice, but, again, really not that exciting to me, right? When you have something that has such a general kind of robust impact on both primary tumor growth and metastasis, I told Aparna, here was a student doing these experiments, there's really nothing exciting here to us. And so Aparna went back to the literature and came up with this hypothesis that TGM2 might be facilitating placement or cross-linking of fibronectin onto these extracellular vesicles, right? And so extracellular vesicles produced by every cell in the body, but cancer cells, based on aberrant changes in gene expression and other mechanisms, start to produce extracellular vesicles that are capable of altering niches around the body, right? And so this is very exciting in that you have a primary tumor can be putting off packets of information and communicating with metastatic sites essentially in an endocrine fashion. And so, definitely some data to suggest that fibronectin as well as TGM2 could be present on extracellular vesicles. And so Aparna did a nice job of isolating vesicles, which is fraught with peril if you go down this road. But very nicely characterized them by nanosite imaging as well as electron microscopy here to show that we have nice vesicle preps. If she does that, she can do Western blots to show that indeed TGM2 is responsible for cross-linking fibronectin on vesicles. So these are Western blots from vesicle preps in which TGM2 is knocked down or overexpressed. And so here again in the BMs, we knock it down. And so if you look at fibronectin, typically if you run a reducing gel, fibronectin is secreted in the matrix as a disulfide dimer, right? And so if you run a reducing page gel, that will reduce and show up as the monomer. So if you do that and you see dimers, that's a good indication that those molecules have been physically cross-linked and run as that dimer under reducing conditions. And so if we do that, we can see reduction in the dimer formation with TGM2 knockdown. We can see induction of the dimer with TGM2 overexpression in these cells. And so if we just stain these, it's probably hard to see on here, but this is an important experiment. Fibronectin is kind of a misnomer in vesicle preparations that it can co-isolate with these vesicles. And so here we're just showing the presence of fibronectin actually on the vesicle, which was an important conclusion for us. And so overall, the hypothesis here is that these primary tumors, again, are putting off vesicles or cells that have disseminated, right, and are in one location might start putting off these vesicles based on TGM2 expression, in which they have TGM2 present, again, with cross-linked fibronectin, which we think occurs on the surface of that vesicle, right? So now it's very exciting in that you have a vesicle that's essentially a packet of matrix proteins that's able to circulate through the body and influence these sites of metastasis. And so to look a bit further in this, we've collaborated with Luis Solorio at Purdue. And so Luis is a biomedical engineer that does a number of things, and one of them which is to make these cell scaffolds. And so if you take these are grids that sit up above the normal tissue culture dish, and so you can do a number of things with these in terms of coating them with matrix or growing tumors on them. But here we're growing human pulmonary fibroblasts, and so we can grow them up in which they start to grow on the polymeric grid, but then cover the open space. So now you have cells that are not supported by any polymer. So essentially a 3D culture that we've compressed into more of a two-dimensional matrix. And so when we do that, we can pre-treat them with the vesicles or not. And so if we do that and then add on just a responder tumor cell line, in this case the CA1As is a derivative of the MCF10A cell line. If we do that and we pre-treat the fibroblasts with vesicles, we can very nicely enhance the growth of those tumors. And then if we knock down or inhibit TGM2 activity, we can block that ability of those fibroblasts to support the growth of tumor cells. And so finally, the in vivo experiment in which you isolate vesicles every other day and treat mice every other day for 3 weeks. So here, Aparna is injecting either PBS or vesicles that have been isolated from those BM cells, either expressing or knocked down for TGM2. And then again, after 3-week pretreatment with those vesicles, we inject those CA1A cells again. And this is a nice model that we knew takes several weeks to grow out in the lungs, and that's drastically accelerated in mice that have been pretreated with these vesicles, and very much dependent on TGM2 expression. And so with that, I'll just conclude saying that we can target a mesenchymal population with FGFR inhibitors. We can inhibit reversion with fostamatinib, which we think might be a very interesting approach to try to prolong or prevent metastatic recurrence. And then for targeting that secondary epithelial population, we're very interested in TGM2, both as a biomarker as well as developing novel chemistry around inhibiting TGM2 activity. And so I'll just thank the members of the lab Again, I tried to call them out as we saw their data. Saeed is very instrumental in lab. My senior graduate student now worked very nicely with Connor, as well as a secondary undergraduate student, Gian, who has recently started graduate school at Baylor. Parna's moved on to a postdoc at AbbVie. And I mentioned Sebastian is a newer student in the lab who I showed some of his data. And all of our collaborators, especially Luis, is great to collaborate with. Tim Ratliff is our Cancer Center director. It's really helping us out with some of the immunology stuff. And then Bob Galen is my mentor in the department that discovered spleen tyrosine kinase and has really kind of guided some of those experiments from afar. He's retired now. And thank you to the funding. Couldn't do it without the financial support. And so I'll take any questions. Haida.
Hi, that was a great seminar. I have a number of questions for you, but
I bet you do.
so one of my questions was, in most of the experiments where you showed sort of this reversible EMT with TGF-beta versus the non-reversible with lopatinib, one experiment looked like when you added TGF-beta was almost all mesenchymal, but in most experiments it kind of looked like you had a mixture when you added TGF-beta, whereas you don't have that mixture when you add lopatinib.
Right.
So I have two questions that are sort of associated with that. Do you know, if you isolate the mesenchymal population like you did to look at gene expression, do those not reverse, or is it the situation where if you're 50-50, let's say, in a dish, and you take out TGF-beta, now the epithelial cells grow better and overcome, and your reversion is actually not a plasticity, but your outgrowth of your epithelial population? That's the first question. And then the second question is, you also showed that you lost HER2 expression in response to
TDM1.
Yes, exactly. And I'm wondering there, even though I know that you expressed HER2 in those HMLE cells, did you ever look just by ICC to see that it was equally expressed in the epithelial versus the mesenchymal compartment? Or could it be, again, that you're selecting for differential expression that is already existing, not that you're actually creating the pressure to change it? Does that make sense?
Yep. So all key questions. I'll do the second one first. In vitro, which that experiment that I showed is in vitro, we have sorted for HER2 expression. All of those cells are HER2 positive. So we don't think we're selecting out for a HER2 negative population. What Saeed's seen there is following EMT, there is a change in trastuzumab binding, right? So all the cells are still HER2 positive using an intracellular antibody. But if you do flow using trastuzumab, following EMT, there's a population that emerges that can't bind trastuzumab. So whether or not that's related to the eventual selection of a HER2 negative cell line, I don't know. But I don't think we're just selecting for a cell line that never expressed HER2. In vivo, it's a little bit different. HER2 expression is much more diverse in vivo, but not necessarily related to epithelial or mesenchymal status. The first question around EMT, we were very much in tune with all of those when we did that study. And so if we sort for the very most 24 negative, 44 high population and put those cells back into culture, we see the same thing, that they are definitely plastic. They definitely return. We do the same thing with the lopatinib cells. They, again, maintain mesenchymal. So we do think it's definitely plasticity and not outgrowth of a population there. And so that one experiment that I showed, I know that is confusing. That was right after TGF-beta treatment. So when you treat with TGF-beta and don't sort, or if you do sort, you get a population that's almost 100% mesenchymal, right? It's a 4-week treatment over trypsinization and stuff like that. But again, we get the mixed population when we remove the TGF-beta and then subsequently culture after that, and we see emergence of the epithelial cells.
Have you considered the innate immune response in the EMT? Because in humans, in vivo, you do the excisional kind of human. That's what induces the immune response, not innate automatic interference. And then I have one looking one DNF, looking 6, and that can promote the lymphatic. So, are you trying to block the innate? You have to do another protection. What about biopsy? And you can block it, you give us an anti-inflammatory. What is when you have the already metastasis, that preventing metastasis?
Yeah, we haven't done too much around innate immunity, primarily because, that 4T07 model, we see this really big difference in metastatic potential in, nude mice versus BALB/C mice, for instance, right? So we've really been focused on adaptive immunity there and the role of CD8s. But no doubt, I'm sure everything that you said there, I totally agree with in terms of a pro-inflammatory microenvironment driving metastasis. We have some projects that I didn't mention here around obesity and actually alcohol intake that are very much along those tracks in terms of altering a pro-inflammatory microenvironment. And how does that change, EMT? How does that change response to therapeutics targeting the adaptive immune system? Yep. Tracy.
My question is somewhat similar to Heida's, but I was curious about your TDM1R cells. So if you switch them to this mesenchymal phenotype, so presumably they're no longer very proliferative.
Yeah, I mean, in general, more mesenchymal cells tend to proliferate slower.
Yeah.
So they definitely proliferate slower than like parental cells.
So they do proliferate. So my question is, if you give them and some kind of proliferative stimulus that doesn't provide ongoing EGFR signal, can they then return to a more proliferative?
Oh, that's interesting. What has he done? He's overexpressed. He's treated those TDM1 resistant cells with FGF, and that does increase their proliferation, but we don't see any kind of reversion around EMT or a reversion back to an epithelial cells there. So I see the concept is interesting. If you drive them to a proliferative state, is that dominant in terms of driving an epithelial phenotype? That's interesting. We haven't approached it from that aspect very much. No. Yeah. A lot more of that paper was taking the parental cells and just overexpressing FGFR to parse out what about FGFR expression is sufficient to drive resistance to TDM1.
When you look at the FGFR inhibitor targeting mesenchymal cells and what the clinical trial would look like. Because based on the EMT, MET model, the primary and the overt mets are going to be epithelial and not respond to your therapy. So by RECIST criteria, you'd have no response to with the combined checkpoint. So how do you actually do that trial?
Yeah. So that's a good point. I think clinically a lot of the FGFR inhibitors are being driven by like HER2 kind of mechanisms in which you're looking for FGFR1 amplification, or like I mentioned, those two other cancers that have FGFR mutations. So, in a typical EMT reversion scenario, when would be the best time to apply FGFR inhibitors? These are very kind of difficult questions. So this is kind of where there are some models in which we've seen FGFR falls into more of that class that it stays elevated following EMT. So following an EMT, if cells upregulate FGFR very potently, they can remain dependent on that. So I think that drives selection for higher level FGFR, even as cells start to move back towards an epithelial phenotype. But if you're looking at, again, all of these things around clinical patients are taking place in a spectrum, right? So you're going to have cells that are in that mesenchymal state, even though you might have actively growing metastases that have transitioned back. And so would it be part of a combination to target that transitioning population as well as the actively growing epithelial cells? But yeah, definitely not. FGFR is a single agent in breast cancer. It's not going to happen,
I don't think you got into this, but this relates back to Heidi's question, but with the TGM2 being activated specifically post-event,
Yeah.
do you look at the mechanism? I mean, is that an epigenetic change in focus? Is it mutational amplification, blah, blah, blah?
Yeah. This is an AIM-3. We don't know how that's working. Traditionally, gene expression changes around EMT have been these kind of master regulator transcription factors, right? So what drives changes in gene expression around these things after EMT's resolved itself? Don't know.
So related to James' question, you identified the TGM2 on your bone metastasis-derived cells, but you tested them on your non-metastasis model. Is this something that is common to all metastatic sites, or do you think that they could be a organ-specific study?
Yeah, I know the way I presented it, it's very kind of compartmentalized in terms of now. So these cells are bone metastasis. They've upregulated TGM2. I try to think about probably more conceptually in which, cells undergo EMT and MET in all kind of locations through the body. And so that's something we're interested in. Is upregulation of this signature important because we pulled those cells out of the bone? Or is it just more related that cells have undergone EMT and then reverted, right? Because at some point, all of those cells, those metastases were derived from a mixed population. But again, all of those cells were mesenchymal at one point. And so I think now we maybe have to go back and just from that mixed population, a population we've never pulled out are of the secondary epithelial cells from just in vitro culture. And so I think we might have to do that to really parse that question apart of whether or not TGM2 or that signature is more related to a bone microenvironment or is it more related to the secondary epithelial state.
I guess related to that is that have you looked at the expression at the metastatic sites in your models?
Yeah, no, so that is, again, in terms of a metastatic model, that's not a powerful metastatic model. We were able to identify a couple of mice and isolate those cells from a couple of mets, but we weren't able to, we're not able to reproduce that on a robust kind of fashion. Yep. I have one back there. She's had her hand up the whole time.
Respect the primary tumor causes an innate immune response. I'm just curious as to whether you've seen this phenomenon. We've seen just the cancer cells even in vitro, and then another group has published some of this too. But when they undergo EMT, they start to show a signature that has commonalities with the innate immune response, like a immune response.
Okay.
So I think it's actually part of the EMT, and I'm sure that the inflammatory environment caused by the surgery can also induce this. My question is whether you've seen that signature coming out before in your cells.
We haven't seen too many kind of pro-inflammatory things. I'm trying to think of an RNA-seq that would recapitulate that. Well, I guess that's not true. When, I mentioned that we were looking at differences in those gene expression patterns between a reversible and a non-reversible EMT. Interferon signaling was the top hit in terms of a signaling pathway that was associated with a reversible EMT. And so in terms of, EMT reversion or stability, I'm not sure what that means, but it could definitely play a role in terms of driving metastatic progression. Yeah, that's definitely true. But in terms of, back to the resection concept, the 4T1s, you can cut those tumors off or leave them on. Mets go forward at the same kind of rate. You can cut the tumors off as soon as they're palpable, and you'll get Mets. We get Mets at the same rate. You can let the tumors get really big and cut them off. So their resection, at least in that model, doesn't seem to affect metastatic progression. I know in some models, I've had a little bit of experience, and I've heard in some models like you need to resect to kind of drive metastasis, but 4T1s, they just plow their way forward. Yes.
So would you project that pathological fibrosis of the lungs might predispose to successful colonization?
Oh, definitely. Jeff Green has done some nice work on this, where they have I think it's an adenovirus, where they induce fibrosis in the lungs. And there's some other models. This D2OR model is a very nice dormancy model, where unlike the 4T07s, you can inject them in the tail vein, and you get very stable disease that doesn't grow out. But if he induces fibrosis in that model, you see very nice outgrowth of the lung. And so that's actually part of that alcohol project is around fibrosis. So alcohol is known to induce fibrosis in the liver and the lungs. So it can that be responsible for inducing the outgrowth of disseminated but dormant cells. Yeah, definitely. So again, this concept of a primary tumor or a disseminated reservoir of cells being able to essentially transmit matrix to different sites, and if that's going to build up somewhere, that's obviously going to be a niche for metastatic bone.
It's a little difficult to dissociate the inflammatory process with adenovirus infection or how the
Sure, yeah.
fibrosis is induced from just the process of fibrosis itself.
Yeah. Right. All right, thanks a lot.